Hey everyone, it's Dr. Marcon, and this is the introduction to Lab 27 on renal function. So Lab 27 focuses on these labs for renal function. We are going over renal regulation. There will be some calculations and concepts that you need to go over for the lab quiz, and then FOX 9.3 deals with the clinical examination of urine, basically how we do urinalysis. Again, as with blood, we do have safety procedures. With urine, please go over, when working with urine, you of course are using universal precautions. Always assume all material is hazardous and make sure you are handling your own urine. And then, please read on the rules for proper disposal of clean or properly dispose of or clean contaminated material. You can actually rinse urine down the sink, but make sure you use large amounts of water. As always, soft contaminated material goes in the biohazard bag. No other trash will go in this bag. Reusable contaminated equipment, wash with Amphil. This includes your test tubes, beakers, pipettes, and slides, and do not wash urine contaminated equipment for anyone else. Again, there is no eating or drinking if we were in real lab, even water. So, if you needed to, you could step out in the hallway, but this doesn't apply to us. So the first part of the lab is FOX 9.1 on renal regulation. So it focuses on renal regulation of fluid and electrolyte balance. So in lab, everyone is expected to go fill up a sample cup, so pee in a cup. The more samples we get, the better. I used to teach medical assisting, and I would tell my medical assistants that they have to properly instruct the patient on obtaining a midstream clean catch sample, and that usually involves telling them how to use towelettes and how to wipe the area around the urethral orifice, using a front to back motion so that we can kind of clean off any cells or bacteria that might be in the area so that we don't have a contaminated sample. So I'm assuming that you guys will be instructed on this had you been in the lab, but anyways, we want to get a good urine sample. A midstream clean catch, basically you pee a little bit into the toilet and then you pee the rest into the cup. So what we're looking for, what we're measuring are these things. We are measuring volume, and there are lines on the urine cup to help us measure volume. We are measuring pH with a pH strip. That involves just sticking the strip into the sample and comparing the color with a standard. And then we are also measuring specific gravity. Now, specific gravity is the ratio of the mass of a substance, in this case the solutes in urine, to the mass of an equal volume of distilled water. We know that the specific gravity of distilled water is 1, meaning there is nothing in water, just water. Whereas in urine, there's going to be stuff in urine. There's going to be, all sorts of solutes in urine, waste products that make urine denser than water. So specific gravity, we're going to be using a instrument called a refractometer, and we will add urine to the blue strip of glass here, closing the top, being careful not to slam the top onto the refractometer. We hold to the light and then look and record the specific gravity indicator. Usually normal specific gravity is anywhere between 1.003, which is indicative of a dilute urine, because it's very close to 1. We know there's nothing in water, so distilled water specific gravity is 1. Or it can be up to 1.040, meaning there this might be a more concentrated urine with more stuff in it. So we use the refractometer to help us determine specific gravity. Once we are finished with this instrument, we clean with chem wipes and of course put the chem wipes in the biohazard container. We're also checking for the concentration of chloride, and by doing this, we want to place 10 drops of urine into a test tube along with 1 drop of potassium chromate. We mix this up and then slowly we'll add silver nitrate. We add it 1 drop at a time and swirl. Now, usually adding the silver will create a rust color that disappears. We want to keep adding drops until the rust color remains and doesn't disappear. And then we want to make sure we count the number of drops it took to turn urine a rust color. Also important to note that potassium chromate is actually the same color as urine. It's a yellowish color. So we're basically looking for the reaction of the silver nitrate into the urine plus the potassium chromate. So again, we count the drops, the number of drops it took adding silver nitrate to the solution, and then we multiply this by 68. We could also multiply by 61 or 66. Now that just depends on the concentration of the silver nitrate we have in lab that day. Now note this is, it's usually a very small percentage difference, so it doesn't really matter too much what factor we multiply by. But again, it's dependent upon the concentration of silver nitrate that is being used in lab. So volume, of course, urine volume will vary depending on what we call void interval time. Someone going to the bathroom 10 minutes ago versus someone who had gone to the bathroom like 4 hours ago. Of course, the one who had just gone to the bathroom will probably not have as much volume as someone who hasn't gone to the bathroom in about 4 hours. So basically, void interval time takes into account how long it has been since emptying the bladder. And of course, this time does need to be recorded. Typical urine volume is anywhere between 20 to 200 milliliters. For pH, average pH is about 6. And the normal range could be anywhere from 5 being slightly acidic, slightly more acidic, to basic at 8. Anything outside this range we want to consider maybe some sort of underlying condition that could be affecting pH. Specific gravity, again, normal range could be a dilute urine at 1.003 to a more concentrated urine at 1.04. And then chloride concentration, it's a wide range. It can be controlled independently or controlled by other ions. Basically, we use chloride to help us stay within homeostasis. We use chloride ions for different things. The body does need to balance out charges and will release negative ions with positive ions to maintain equilibrium. So there can be a huge range for the normal range. So again, a wide range can vary from anywhere from 1 to 40 drops. So when we do multiply that, we get a number from anywhere of 68 to 2,720 milligrams per deciliter for the concentration of chloride. And we'll get into why we are measuring the concentration of chloride in the discussion. So this should be 9.3, the clinical examination of urine. I used to tell my my medical assisting students that we not only are doing the chem strips to test for these things within urine, but we also want to do kind of a physical examination of the urine. We want to take a look at it. We want to observe, volume as well as color. Is it a light color or a darker color? Is it fairly translucent or is it opaque? Does it look like there's a lot of, solvents or solutes in the urine? So that's sort of how we look at it doing a physical examination. Also, we take note of any odor that the urine might have, and we record this down in the patient's chart. So we know that color of urine is a good indication of hydration status. If someone is regularly drinking water and keeping themselves well hydrated, their urine is probably going to be a lot paler in comparison to someone who is severely dehydrated and hasn't drank water in a long time. And they'll have a more darker color, and their urine won't be as pale or translucent. So again, urine being a really good indicator of hydration status. So for the lab for clinical examination, basically we are taking a chem strip, and you can either spray urine onto the strip, or what I usually tell my students to do is to dip the chem strip and then dab off any excess urine from the chem strip. And at that point, the chem strip will have several indicators. Sometimes they'll just have one or two, but for the most part, universally, they'll have about 5 indicators for protein. PRO stands for protein, GLU will stand for glucose, KET stands for ketones, BLO stands for blood, and BIL will stand for bilirubin. So the chem strip is actually very time sensitive, and you have to compare the strip to the bottle, and the bottle will tell you at what time you have to read the colors or the color changes on the strip. And it's usually good to make sure at what time you have to read the color indicator or the color change, because if it stands around for a really long time, you might not get accurate results. So make sure you have a timer with you while you are comparing the chem strip to the colors on the bottle. The next part of this experiment is doing a urine sediments exam. So basically we have a centrifuge tube or a plastic tube that we fill halfway with urine. We then will centrifuge this plastic tube for about 5 minutes. Make sure you are balancing your tube. And then what it does is usually any sediments that might be in the urine will fall to the bottom of the tube, and you pour the urine that's at the top into the sink. You then add what's called a SETI stain. This is a dark purple mix, and you want to mix this by aspiration. So basically you take a pipette, place it into the SETI stain, and then place that into your little plastic tube with the remaining sediments, and then you mix by aspiration by sucking whatever's in the tube back up into the pipette and spitting it back out to mix the sediment with the SETI stain. And then you add this to a slide. You'll be able to, if there are any sediments within or any crystallizations or anything in the sediments, you will be able to see this under a microscope. Now, most of the time, students come out disappointed because they don't see any sediments. And you know what? Actually, that's a good thing, because sediments in your urine might be indicative of some sort of pathological condition in your kidneys, and if you guys are testing your own urine, you kind of don't want to see the sediments or anything underneath the microscope. But, however, you can look at your book to see, examples of some things that you might find under this stain. So that is Lab 27 and all the activities that you would have performed in this lab. Next part will be the discussion on renal function in this lab.